Hello, my name is Lauren McElroy, and I will be an incoming freshman at RIT this fall. And first and foremost, I would like to apologize for any background noise. There's some work going on in my apartment complex, so I'm just going to get started. Over the past 4 weeks, I have been working with Dr. Krista Wadsworth on the RIT Fast Forward Summer Research Program. The project I have been working on involves researching the causal agents of antibiotic resistance in the pathogen Neisseria gonorrhoeae, which causes gonorrhea. Neisseria gonorrhoeae is a causative agent of the sexually transmitted disease gonorrhea. Gonorrhea causes a wide range of symptoms that vary in severity. These symptoms include frequent urination, fever, persistent sore throat, cervicitis, and urethritis. Rate of infection, as well as more severe complications from gonorrhea, are rapidly increasing. Not only that, but gonorrhea is becoming more and more resistant to the antibiotics that are used to treat it. This graph depicts the increasing rate of infection since the year 2008 and compares the infection rates between males and females and the difference between how fast the infection rates are increasing between them. This graph shows how gonorrhea has become increasingly resistant to different forms of the antibiotics that are used to treat it since the year 2001. As you can see, the antibiotics that gonorrhea is becoming most resistant to faster are tetracycline, penicillin, and ciprofloxacin, whereas azithromycin, cefixime, and ceftriaxone are more effective treatments, and I apologize if I didn't get those names right. This image shows where the different species of Neisseria exist in the mouth. The human body actually contains a lot of different species of Neisseria, but they're not all Neisseria gonorrhoeae. So some of these species that are in our body contain the mutations that are necessary to cause antibiotic resistance, even though they don't cause gonorrhea themselves. However, gene sharing is very common, and so the different species of Neisseria can basically give each other the genetic information necessary to give them the antibiotic resistance. So the antibiotic resistance in Neisseria gonorrhoeae is coming from that of another species that is not related to gonorrhea. This table shows the individual isolates that I worked with and their minimum inhibitory concentrations, or MICs, of each antibiotic treatment. The isolates are not all the same species of Neisseria, so this table helps to show where the resistance may come from and was produced at RIT by Jordan Raisman and McElroy. The sequences shown on this table were produced at the Genomics Core faculty at RIT by Neri and Wang. The table shows each strain their genome assembly sizes. All genomes had roughly one gene per 1,000 base pairs. The goal of this project was to analyze the isolates and find the most probable gene mutations that cause antibiotic resistance in Neisseria gonorrhoeae. Identifying these causal agents is important in the development of new treatments that help stop the spread of gonorrhea, as well as finding a way to slow down and stop the increasing resistance to antibiotics in Neisseria gonorrhoeae. In order to analyze the isolates, I put them through the Comprehensive Antibiotic Resistance Database, or CARD, from the CDC. This database separates the genes that are known to have been linked to antibiotic resistance and their SNPs, or single nucleotide polymorphisms. If the gene had a SNP, then that means that it had a mutation. I took these results and I placed them into a spreadsheet separated by the individual isolates so that I could compare the results between them. In my results, there were several mutations that appeared on every single isolate. Their common presence implies that these mutations are not likely the cause of resistance in these isolates. So I took these mutations out of my data so that I can narrow down my results into a more comprehensive list of options. The list I ended with consisted of 8 mutations that each appeared 6 or fewer times, and by that I really mean that only one of them appeared 6 times and most of them were much less frequent than that. In fact, 50% of this list only showed up once or twice. This table displays my results. The yellow cells are containing mutations that are less common, but still not as likely to be responsible for antibiotic resistance, so they showed up fewer than every single isolate, but they still showed up enough that I don't feel confident enough to say that they are likely the cause of antibiotic resistance. However, the green cells indicate the isolates containing the 8 mutations that I found most probable to be the causal agents of resistance. So those 8 mutated genes that I had found and listed out are the ones that are in the green cells. I compared my table from the previous slide to that of Tatum D. Mortimer and Yonatan H. Grad from their paper Applications of Genomics to Slow the Spread of Multi-Drug Resistant Neisseria gonorrhoeae. This paper contains a similar table containing some of the same genes I had found in my own research. However, while some of the genes were the same, the SNPs did not all match up. The genes I found with the same mutations on this table as there was on mine were the MTRR and RPSJ genes. These were included on my list of 8 genes most likely to be responsible for the resistance to antibiotics. So these results show me that those 2 genes are the most likely to be responsible for the mutations causing antibiotic resistance in the Neisseria species. And finally, I would like to acknowledge RIT and the Fast Forward Summer Research Program for the amazing opportunity to work on this project. I would like to acknowledge Sandy Connolly for organizing it and Dr. Krista Wadsworth for guiding me through the research. I would also like to acknowledge the Comprehensive Antibiotic Resistance Database for making my life 100 times easier during this process, and Christy Lutzer, the Senior Staff Specialist, for also helping to keep everything on track during this program. Thank you very much for watching.